Okay, second year MBBS class. We was started our new topic of biochemical genetics, and today we are going to start DNA replication. So DNA replication is basically the replication or the doubling of the DNA. Basically, it is the basis for inheritance, and it's a fundamental process occurring in all cells for copying DNA to transfer the genetic information to daughter cells. The primary function of DNA replication is understood to be the provision of progeny with the genetic information possessed by the parent. So, the replication of DNA must be complete and carried out in such a way as to maintain genetic stability within the organism and the species. The process of DNA replication is complex and involves many cellular functions and several verification procedures to ensure fidelity in replication. For about 30 proteins are involved in the replication of E. coli chromosomes, and this process is almost certainly more complex in eukaryotic organisms. The first enzymologic observation of DNA replication is the replication of the DNA. So, in all cells, replication can occur in stepwise manner. These steps of DNA replication may include the same steps in prokaryotes and eukaryotes, but the components or the proteins involved in these processes would be different. So each cell must replicate its DNA before division, and all the processes which are involved for the replication of the DNA, these could be the identification of the origin of replication, it's the unwinding or denaturation of double-stranded DNA, the formation of replication forks, initiation of DNA synthesis, then elongation, and ultimately the termination, both in case of prokaryotes as well as eukaryotes. But to go into the details of DNA replication, we must know some prerequisites for the DNA replication. First of all, DNA replication is a semi-conservative method in which half of the DNA is conserved and half become available for the synthesis of the new strand. Parental strands are not degraded. Base pairing allows each strand to serve as a template for the new strand. New duplex is half parent and half of the template, as it's a semi-conservative. So the DNA replication, in this, the energy for the DNA replication arrives from the phosphate, which are the DNA bases and sugar attachments. DNA bases arrive with their own energy sources for bonding, and they are going to bond first of all with the DNA polymerases. So for the start of the DNA replication, a primer is needed to be attached or identify to the origin of replication. A primer is a DNA primer can attach with the DNA polymerase, and it can only add nucleotides to the 3' end of the growing DNA strand. So DNA polymerase for its working needs a primer, and a primer could also be called as a starter. So DNA polymerase need a starter called primer for the synthesis of the daughter strand. Strands could only move or grows in 5' to 3' direction, and the template is read in the 3' to 5' direction while polymerization takes place in the 5' to 3' direction. As you all must have an idea about the Okazaki fragments, the leading strand and the lagging strands. So this is the initiation of the formation of those leading and lagging strands. Like in DNA replication, one strand is going to be made in the 3' to 5' and the other will be in the opposite direction. But the DNA polymerase could only work in the 5' to 3' direction. That's why the template strand must be in fragments. The synthesis of the template strand or the daughter strand must be in fragments. So what is a primer? The primer is always single stranded, so it is called the RNA primer. It is synthesized by primase enzyme. It serves as a starter sequence for DNA polymerase 3. Only one RNA primer required for the leading strand. RNA primers for the lagging strand depend on the number of Okazaki fragments. So RNA primer has a free 3' hydroxyl group to which the first nucleotide is bound. These are the overall steps for the DNA replication. First of all, the identification of origins where the replication is going to be started. Then the unwinding or the denaturation of the double-stranded DNA to provide RNA primer a single-stranded DNA template. After the denaturation or unwinding, a replication fork is going to be formed. After which the RNA primer starts or initiates the DNA synthesis and elongation. Afterwards, primer is going to be removed and ligation of the newly synthesized DNA segments occurs. And at the end, the reconstitution of the chromatin structure takes place, which is the again winding of the DNA with the doubling of that specific part of the DNA that is required for the synthesis of specific protein. So to follow or to accomplish the DNA replication steps, we have different components of replication. These components includes DNA polymerases, helicases, topoisomerases, RNA primase, single-stranded binding proteins, and DNA ligases. For DNA polymerases, what is the function of DNA polymerase? It is going to do polymerization, as the name indicates. Helicases are for unwinding of the DNA. During unwinding, DNA encounter many supercoils. To relieve those torsional strains or supercoils results from the helicases activity, we need topoisomerases. We will study the function and the functioning of topoisomerases afterwards. Then comes the RNA primase, which is going to make or synthesize RNA primer. Then single-stranded binding protein are also there, which are going to bind with the single-stranded RNA to prevent the premature annealing of the double-stranded DNA. And finally, for the ligation or for the seals or for the sealing of the single-stranded DNA, we need DNA ligases. And these DNA ligases must function both in leading and lagging strands. So what is the origin of replication? Origin of replication is called ORIC in case of prokaryotes. There is an association of sequence-specific double-stranded DNA binding proteins with a series of direct repeat DNA sequences. In E. coli, the OREC is bound by the protein DNA A. So these are the OREC is that origin of replication where the primase is going to be bound, but for the primase to bound with the OREC, we need protein called as DNA A protein. So a complex is formed consisting of 150 to 250 base pairs of DNA and multimers of DNA binding proteins. This leads to the local denaturation and unwinding of the adjacent AT rich region of the DNA. So what is the concept behind this is that first of all, the origin of replication is identified by the primer, and that origin of replication is specifically rich with the A and T base pairs, where DNA A protein is bound, which is capable to bind with the RNA primer. But there are different sequences present in case of eukaryotes other than AT rich region. These are called autonomously replicated sequences or replicators. For example, in yeast cells, the ARS contains a somewhat degenerate 11 base pair sequences called origin replication element. Now we are talking about eukaryotes in yeast cells. The ORE binds a set of proteins analogs to the DNA A protein of the E. coli, which is collectively called as origin recognition complex. So what we call origin of replication OREC in eukaryotes will be called as ORC in case of eukaryotes. The ORE is located adjacent to an approximately 80 base paired AT rich sequences that is easily unwinded. This is called DNA unwinding element. So these are the exceptions in eukaryotic cells with prokaryotes. After the identification of origin of replication, the DNA is going to unwind. The interaction of protein with OREC define the start site of the replication and provides a short region of single-stranded DNA, which is essential for the initiation of synthesis of the nascent DNA strand. So now unwinding is attached with the helicases. As the DNA unwinds, DNA helicases moves forward and causes the DNA to become unwired. DNA helicases allow for the processive unwinding of the DNA. And as the DNA unwind, single-stranded binding proteins go and attach with the single-stranded DNA. In cooperation with SSB proteins, this leads to DNA unwinding and active replication. This is how the origin of replication is identified, as well as the replication fork is formed with the help of DNA helicase enzyme. After the movement of helicases, there is the formation of replication fork. This diagram is from Lippincott, and this is called as the elongation step of the replication, as the initiation is done with the help of in the identification of origin of replication and the primer binding. Afterwards, we will move towards the second step of replication, which is called as elongation. Then after it, we will end this process of replication with the termination. So what happened in the replication fork formation? The polymerase 3 holoenzyme binds to template DNA as a part of multi-protein complex. So here you can see in green colored the DNA polymerase. During the initiation, helicase the triangular green color which is going to be moved forward is the helicase enzyme, and the egg-shaped yellow colored substances are the single-stranded binding proteins. And in this step of replication elongation, DNA polymerase is going to bind and move in the 5' to 3' direction. As always keep in mind, DNA polymerase always move only in the 5' to 3' direction. So the DNA polymerase is only able to move in the 5' to 3' direction, and because of the DNA strands are antiparallel, the polymerase function asymmetrically. Like I was telling you before, the one strand would be called as the leading. The forward strand, the DNA is synthesized continuously because the function of the DNA polymerase is only to move in the which direction? The 5' to 3 direction. But what happened on the antiparallel, the other strand, the lagging strand, the retrograde strand, the DNA is synthesized in the form of small fragments. Those fragments could be in the 1 to 5 kb in size, and these would be called as the Okazaki fragments. Here you can see the formation of Okazaki fragments in the lower strand, where the DNA polymerase cannot move continuously and results in the formation of small fragments, which afterwards are going to be ligated with the help of DNA ligases. So the important things in this diagram is the initiation, which is done with the help of RNA primer, then the role of helicases and single stranded binding proteins, and during elongation DNA polymerase is going to function in the continuous and discontinuous manner. During the continuous manner, it will result in the formation of leading strand template, while the lagging strand are going to be formed with the help of different Okazaki fragments. So this results in the formation of many replication bubbles in this way, specifically in the case of E. coli. So as you know, in E. coli there are two types of chromosomes. One is the chromosomal DNA and the other is the extra chromosomal. And in E. coli we always talk about the extra chromosomal DNA in the A part of this diagram, where the circular DNA is called as a plasmid, and only one replication bubble is going to be formed and only one replication fork is formed. While in case of eukaryotic DNA, multiple origin of replications are there, and which results in the multiple replication bubbles, which will go and cause the replication of multiple areas of the DNA. So replication occurs in both directions along the length of the DNA, and both strands are replicated simultaneously. The replication process generates replication bubbles. Here you can see the replication bubbles both in prokaryotic and eukaryotic cells. So during this replication process, a DNA polymerase complex is formed. What are these? The chain elongation, the processivity, and the proofreading. During chain elongation, it accounts for the polymerization rate. Processivity is the elongation, and proofreading is the correction. First of all, the identification of the error and then the correction. So these all steps, including in DNA replication process, completes the process of DNA replication, where we will end the process of DNA replication by correcting the errors, which are called as different mechanisms of DNA repairs. In E. coli, DNA polymerase 3 function at the replication fork of the polymerases. It catalyzes the highest rate of chain elongation and is the most processive. Polymerase 2 is mostly involved in proofreading and DNA repair, and 1 completes the Okazaki fragments and lagging strand. Here you can see some differences between polymerase 1, 2 and 3. We will discuss in detail in the next lecture, during which we will discuss the function of topoisomerases, and there are some differences between prokaryotic and eukaryotic polymerases also. Like in E. coli, the polymerase 1 function as a gap filling and synthesis of lagging strand. Same function is done in the mammalian cells called as the alpha polymerase, while 2 would be replaced with the name epsilon in case of mammalian cell, and 3 will be with the delta. And in E. coli we will encounter 3 types of polymerases, where DNA polymerase 3 is the most important and involves in polymerization, while 2 and 1 are involved for the proofreading activity. While in mammalian cells, the polymerization will be done with the help of delta, and gap filling and proofreading and repair will be done with the help of alpha and epsilon. You will keep in mind these names for the MCQ especially. So with this we will end today's lecture. Tomorrow we will be ending this DNA replication along with some concepts or some other helping enzymes called as topoisomerases.